Okay, now we are back and we're going to start with section 14.2, and this is actually going to explain to you how transcription happens. So as you recall, as part of the central dogma, we start with DNA and then we move to RNA, and transcription is that first stage that gets us from the gene starting to get to the protein, but we don't get to the protein by the end of transcription. So you might recognize some terminology, and the first thing you'll probably recognize is something known as RNA polymerase. So similar to DNA polymerase, RNA polymerase plays a crucial role in RNA synthesis, just like DNA polymerase helps with DNA synthesis. So RNA polymerase breaks the two strands of DNA apart and is also responsible for joining complementary RNA nucleotides together. So it's really the driving force behind transcription. And this is what starts our transcription. RNA polymerase is able to assemble polynucleotides in the 5' to 3' direction, but this doesn't cause any problems like in DNA because there's only one strand of RNA, so you're good to go. And also, RNA polymerase can start the chain without a primer. So if you remember with DNA replication, you needed that RNA primer. You don't need that. The transcription is initiated by something known as the promoter, and this is a DNA sequence that RNA polymerase attaches to begin transcription. And as you remember, what happens once it attaches is DNA begins to unwind, and you start to get the RNA. And this first step of transcription is known as initiation. The second step is known as elongation, and that is when our RNA chain is getting longer because RNA polymerase is adding the complementary nucleotides, once again going from the 5' to 3' direction. And it adds nucleotides, boom, 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 to the 3' end of the DNA molecule. And then finally, we have something known as termination, where RNA polymerase comes off and the completed RNA transcript is finished. And in bacteria cells, you will see a terminator similar to the promoter that signals the end of transcription, but that's only in bacteria cells, not eukaryotic cells. So once again, just as a little quick review, RNA polymerase, it attaches to a DNA sequence known as the promoter in both eukaryotic and prokaryotic cells. And in bacteria, there is also an end signaling sequence known as the terminator. Once again, there's 3 stages: initiation, elongation, and termination, and those basically describe what's happening in each one, but I'll go over them one more time very quickly. Keep in mind, this is transcription, is how RNA is synthesized from DNA. Don't forget about that. So with our initiation, we have the promoter signaling the start point, and there are also some things known as transcription factors which help the RNA polymerase bind to the DNA and initiate transcription. And also, when we are taking a look at initiation, this promoter, there's a lot of different promoters that do exist, but one of the most crucial ones is known as the ta-ta box. That's not super important, but you might see that a lot, and it's adenine, thymine, adenine, thymine. These transcription factors will bind to the DNA, and then this transcription initiation complex forms with our RNA polymerase, and that allows the RNA transcript to begin forming. As it moves along the DNA, it will untwist the double helix, which begins to start the elongation of our RNA molecule. And transcription happens very, very quickly. So that's happening very quickly. And then as the RNA polymerase moves along the DNA template, adding nucleotides, once it starts to finish off, it will slowly peel away from the DNA molecule, and then the DNA double helix will reform behind it. So that is one thing that happens in elongation. Once we get to termination, this is where the 2 types of cells deviate paths. So we're going to talk about bacteria first and then eukaryotes. In bacteria, remember, they have a sequence known as the terminator, and that's the sign for RNA polymerase to stop. So transcription will continue until it hits the terminator sequence. The terminator sequence gets transcribed. That's the signal for it to stop. The RNA polymerase detaches from the DNA and it releases the transcript. And remember, because there's no nucleus, the transcript is ready to go straight into the cytoplasm for translation. No extra steps needed. On the other hand, when we talk about eukaryotes, they do not have a terminator per se, but they actually, the signal to stop is known as the polyadenylation signal sequence. So once RNA polymerase transcribes that, it signals the nucleotides to get released, and the RNA transcript is cut from the polymerase, and this releases what's known as pre-mRNA, and that's going to move on to some further processing in the nucleus before it's released into the cytoplasm for translation. And then we'll start on 14.3 in the next video. So hope you really enjoyed that.